We recommend running the loops on a 0.7% agarose gel in 0.5x TBE at 4 volts per centimeter. That's 100 volts in a standard 25 centimeter owl gel box for 100 minutes. It's important to use TBE instead of TAE since the separation of looped from unlooped is significantly better in TBE under these conditions. Other running buffers have not been extensively tested. Other agarose percentages can be used, but we recommend staying between 0.6 and 1%. Adjusting times and voltages to suit your needs is possible, but we have not extensively tested this, so it is recommended to follow the protocol for a baseline before tweaking parameters. To prepare and run the gel, add 0.7 grams of ultra-pure agarose to a 500 ml Erlenmeyer flask. Make 2 liters of 0.5x TBE by diluting 100 ml of 10x TBE with 1900 ml of reverse osmosis, milli-Q, or double distilled water. Add 100 ml of 0.5x TBE with a magnetic stir bar to the Erlenmeyer flask. It is important to use the same buffer for making the gel and for the running buffer. Even slight mismatches in buffer can cause gel artifacts that make quantification difficult. Make sure you have enough 0.5x TBE to make the gel and fill the gel box with running buffer. Cover the top of the flask with aluminum foil to prevent vapor from escaping. Place the Erlenmeyer flask on a hot plate with stirring capability. Bring to a rolling boil while stirring at roughly 400 RPM. Allow it to boil for several seconds. Cool the flask until it is cool enough that it can be handled comfortably in a gloved hand for 60 seconds. If the gel cools too much, it will not pour evenly, and it is better to err on the side of too hot rather than too cold. We find that cooling the gel by running cool water over the flask for about a minute or by submerging it in ice water for 20 seconds tends to provide quick and adequate cooling. Ensure that you swirl the contents while doing this so that the liquid does not gel at the walls of the flask. Pour the gel into a molding tray with a gel comb containing at least 13 wells and allow this to solidify. We find that pouring the gel in the cold room at 4 degrees C helps expedite the gelling process. 10 minutes in the cold room should be ample time. At 25 C, the gel should be allowed to sit for at least 45 minutes to solidify. To each of the 12 microliter samples, containing 2 microliters of biotin and 10 microliters of the construct streptavidin mixture, add 2 microliters of 6x Ficol loading dye. We recommend running a ladder alongside the on-rate samples to aid in verification of the looped and unlooped bands. To prepare the ladder, mix the following: half a microliter of 1 kb extension ladder, 4 microliters of 6x Ficol loading dye, and 19.5 microliters of half-x TBE. Fill the gel box with the 0.5x TBE running buffer to the fill line. Again, remember to use the same buffer used in casting the gel. Load 4 microliters of ladder into the first lane. Load the full 14 microliters of each of the time points into the next 10 lanes. Additionally, 10 microliters of the unquenched sample can be mixed with 2 microliters of 6x Ficol loading dye, and these 12 microliters can also be loaded. In the final lane, load 4 microliters of the ladder mix. Having a ladder on both sides helps if there is skew at the running or imaging stages. Next, run the gel by applying 4 volts per centimeter, with the distance measured from electrode to electrode. Do this for 100 minutes. Note that if the interaction of interest has a lifetime shorter than the gel runtime, the loops will fall apart during the gel running process, and no looped band will be seen. To circumvent this, we have shown separation of the looped and unlooped for a VAR 4.8 construct in as little as 9 minutes at 300 volts. See supplementary figure 4 in the paper. In a container slightly larger than the gel, dilute the SYBR-gold 1 to 10,000 in the running buffer. For example, 6 microliters of stock SYBR-gold in 60 milliliters of 0.5x TBE. We recommend using SYBR-gold for maximum sensitivity when staining the gel. Allow the gel to sit and stain on a rocker for at least 30 minutes. 1 hour usually leads to more even staining. We have found similar results using several imagers, including a point-and-shoot camera. But for the highest sensitivity and repeatability, we recommend using a laser scanning gel imager, such as the Typhoon FLA 9000. Note that some gel imaging formats save with nonlinear intensity profiles. This will affect your analysis, as the intensity will no longer be linearly proportional to the amount of material. Either make sure that you save the images with a linear intensity profile, or that you linearize the images with freely available tools in ImageJ. SYBR-gold can be excited with 488 nanometer light. On the Typhoon FLA 9000, we usually image using the standard SYBR-gold settings with a PMT value of 500, and a resolution set to 100 microns, or 10 microns if we want to resolve two bands that are very close to one another. See our online methods and supplemental for details about image analysis, quantification, and kinetic modeling. This is an image of a gel prepared, run, and stained under these conditions, showing where the looped and unlooped bands appear relative to the 1 kb extension ladder.